My name is Sarah Gora. I work in the Kerner Biodiversity Lab. The title of my research is Legume-rhizobia symbiosis. How do plant traits influence number of nodules in the longleaf pine? So first, what is nitrogen fixation? Nitrogen fixation is when a plant takes atmospheric nitrogen that it can't use and it converts it to a usable form, ammonia, where it can use it. Members of the bean family or the legume family can form symbiosis with rhizobium. This rhizobium bacterium can grow in specialized plant organs called root nodules. Rhizobia fix nitrogen for the legume host, receiving carbon in exchange. So why legumes? No other plant family has been found to compatibly host this unique symbiosis. My research was done in the longleaf pine savanna ecosystem, or the LLP. This was historically the dominant ecosystem of the Southeastern United States. Out of 96 million acres, only 3% of its original habitat is now remaining. Nitrogen fixation can be particularly important in these fire-dependent ecosystems like the LLP, where nitrogen is limited in these frequently burned soils. I have 3 research aims. First, are plant volume or leaf number predictive of biomass? Next, do plant traits predict the nodule number and nitrogen fixation rates? And does nodule number influence the nitrogen fixation rates or below ground resource allocation? I sampled 2 common and dominant legumes of the longleaf pine. I collected 25 plant samples of each species in the Sandhill Gamelands in Hoffman, North Carolina in July of 2020. Table 1 shows the plant traits I collected. In the field, I collected leaf number and plant volume. Whole plant samples were then taken back to the lab, and I weighed them to get the above ground and below ground net primary production. After washing the roots, nodules were then collected and counted. Root photographs were then taken, and they were put into Photoshop, where virtual root processing was then used to find root area and nodule percent pink, which correlates with the nitrogen fixation rate, where a higher percent pink means more nitrogen is getting fixed. I used a 2-tailed t-test to compare single traits between the 2 species. In my graphs, we have Lespedeza hirta in green, and then we have Galactia regularis in yellow. Figure 1 is showing the total number of nodules found on the roots of my 2 legume species. My p-value is 0.161, showing that there was no effective species found in predicting nodule number. Figure 2 is showing that the biomass allocation pattern with a p-value of 0.173. There is no effective species in predicting the biomass allocation. Next, in my third panel, I have linear regression models, and I ran them to find relationships between the plant traits. Again, you have Lespedeza hirta in green with Galactia regularis in yellow. Figure 3a is showing the relationship between the above ground net primary production on the x-axis and the plant volume on the y-axis. As above ground net primary production increases, so does plant volume. In figure 3b, we have the relationship between A and PP and then the leaf number, with leaf number being on the x-axis. As A and PP increases, so does leaf number. In my figure 4a, I have my relationship between the biomass allocation ratio and the number of nodules in my 2 species. Galactia regularis is showing the biomass showed a positive correlation with nodule number, and Lespedeza hirta biomass showed a negative correlation with nodule number. In figure 4b, we are seeing the relationship between the plant volume and the number of nodules, with plant volume and Galactia regularis found to predict nodule number. So to conclude, there was no effective species in predicting the nodule number and fixation rates or biomass allocation. My finding for my first aim was that leaf number and plant volume can predict above ground net primary production. Finding for my second aim is that the plant volume was the only trait with enough support found to predict nodule number. To answer aim 3, there was a significant interaction with species in nodule number with my p-value of 0.04 on the biomass allocation ratio. So looking forward, demonstrating impressive ecological value due to their diversity, abundance, and then their importance to wildlife, The native legumes of the longleaf pine could be of particular interest in conservation as well as restoration efforts because this ecosystem is recovering. Special thanks to the Sandhill Game Lands in Hoppin, North Carolina for letting me use their site. And then thank you to graduate student Alyssa Young and then my advisor, Dr. Kerner, for guidance in this project.